So, good morning. My report is all about metabolic system of insects, particularly the storage and utilization of energy and nutrients in insects' fat body. When we say fat body, it is the central storage depot for excess nutrients. Fat body also plays an essential role in energy storage and utilization. Fat bodies Fat bodies store and release energy demands of insects. Fat body function is not only to control the synthesis and utilization of energy reserves, but also synthesize most hemolymph proteins and circulating metabolites. The fat body function in many aspects of energy storage and synthesis of proteins, lipids, and carbohydrates. The mobilization of these three macronutrients, the lipids and the carbohydrates, is controlled by the adipokinetic family of peptides and the family of insulin-like peptides, or ILPS. When we say adipokinetic hormone, this is produced by the corpus cardiaca, while the insulin-like peptides are produced in the median neurosecretory cells of the brain, the corpus allatum, the corpus cardiaca, and the peripheral tissues, including the fat body. So let's proceed to where the proteins, lipids, and carbohydrates synthesize. So first is the lipids. Lipids is the primary storage molecules and an essential source of energy in insect during reproduction. Lipids are stored as triglycerides. The fat body is the principal storage site of lipids and insects. Most of the lipid is present as triglycerol or triglyceride. When we say triglycerol, these are the main constituent of vegetables and animal fats and diet, and also the main constituent of body's fat stores. Lipids also reserve metabolic energy in insects. Lipids are important because they are more efficient form of energy storage than carbohydrates. So there are two reasons why lipids are more efficient form of energy storage than carbohydrates. First is lipids contain approximately twice the amount of energy per gram than carbohydrates. Second is glycogen contains substantial water of hydration, while lipids droplets contains little or no water. So let's now proceed to the second one, which is the proteins and the amino acid. The fat body is the principal site of synthesis of hemolymph proteins. The fat body also produces diapause proteins. The fat body plays an important role in amino acid metabolism, principally because it is a major site of transmination between amino acid. Proteins in amino acid contributes to the regulation of the amino acid tyrosine content of hemolymph. Proteins in amino acids perform a similar function to other organic compounds. They are also an energy source. Proteins and amino acids have as substrates for other sugar transformation and building blocks for other molecules. So the last one is carbohydrates. Carbohydrate is stored in the fat body as glycogen and circulates in the hemolymph in the form of trehalose. Trehalose is an energy-dependent process or an instant source of energy. Carbohydrates are stored as glycogen polymers of glucose. The amount of stored glycogen in the fat body is determined in part by the labels of trehalose and the hemolymph. Hemolymph is the major transport medium for exchange of material cells such as hormones, waste materials, and nutrients. In carbohydrates, both glycogen and trehalose are synthesized in the fat body from UDP glucose, which is derived from the dietary carbohydrates or amino acids. As the level of trehalose rises, its synthesis in the fat body is inhibited and UDP glucose is diverted to glycogen synthesis. Glycogen serves as a source of cryoprotectants and overwintering insects. Glycogen is an easily activated glucose backup made of glucose residues. Glycogen is located in muscle tissues and can be found also in insects' eggs. Lastly, glycogen is also used to synthesize chitin, which forms the shell of insects. So that's all about my report entitled Metabolic System of Insects, Fat Body, Storage and Utilization of Energy and Nutrients in Insects.